I'm going to talk about, and to some extent would have overlapped a little bit with Marilyn, really about a neurodegenerative disease, ALS, but as well to a larger disease beyond the rare neuromuscular disease and fatal disease ALS, and that's to dementia as a whole. I can't help but to start, and let's see if this runs properly. No sound.
Myself,
there we go.
The luckiest man on the face of the earth. That I might have been given a bad break, but I've got an awful lot to live for. Thank you.
So we often think of this terrible disease in the face of Lou Gehrig in the United States. Of course, it's, in medical terms, it's really known as amyotrophic lateral sclerosis. And it's a disease largely of motor neurons. So the motor neurons are the cells that are a rare population of cells actually in your brain that connect from your brain. They initiate all motor movements. Allow me to move my thumb, press the keys. That's where the initiation of motor movements occurs. And these cells directly connect then to the lower motor neurons, the neurons in your spinal cord and brainstem, the base of your brain, which connect directly to muscle to allow muscles to contract. That simple system of two circuits really allows all voluntary motor systems. It gets refined by other systems in the brain, but that's the core of your voluntary motor system. And you obviously need that. You need it to walk, breathe, talk. And in ALS, there is a death of these cells, both the cortical cells, and the loss of cortical cells actually leads to a clinical phenotype, typically we call spasticity, which is a stiffness of movement of muscles, reflexes that jump quite fast. That's going to hurt. Loss of lower motor neurons, the ones in your spinal cord, lead to the atrophy of your muscles, the thinning of muscles, weakness, and the inability to move muscles. Muscles and motor nerves are very much like a very good marriage. You need that motor neuron connecting to the muscle to work properly. If you remove the lower motor neuron, whatever else you try to do, you move the arm, you stimulate it, you take any kind of supplements you want, it will not matter. That muscle will degenerate. It must have a lower motor neuron. And that's the hallmark, the clinical hallmark of ALS, the loss of those two populations. Now, it's an oversimplification because, in fact, although ALS we think of as a motor systems disease, we now know that about 50% of patients actually have a mild dementia because it's not just the motor neurons that degenerate in ALS, those rare population of cells in the brain and spinal cord, but other neurons in other parts of the brain, very much overlapping with other dementias. And one that I'll talk about is known as frontotemporal dementia. We don't tend to notice the dementia in these patients because the motor neuron disease and the fatality of the disease is so severe that it overrides the perception of what is really often a mild dementia. And often, in the past, we'd often write it off to being patients were depressed. It's a terrible disease. It's understandable that patients are depressed and not perhaps focus as well. But we now know with very good formal testing, and you're going to hear later this afternoon from a colleague of mine, R.G. Hillis, who actually specializes in some elements of dementia, that this is now commonly seen in ALS. So the loss of the motor neurons leads to the inability to do a lot of normal functions. You can't walk properly because the lower motor neurons that control the legs aren't working. You can't drink or write properly, and if you can't swallow, you start losing body weight. But the disease is uniformly fatal because you'll lose the ability to drive your diaphragm. Your diaphragm, the muscle in the lower part of your chest, is critical for taking air in. If you can't take air in, it doesn't matter what you do, you will die. ALS is a uniformly fatal disease, no exceptions. This is not like cancer where there are occasional patients who sort of recover. Maybe they do miraculous things or go somewhere to take some funny drug or that they think is important. ALS doesn't matter. You're going to die, and you're going to die generally in 2 to 5 years. You will live if we put you on a ventilator, and we rarely do that in the United States on patients. When I say rarely, we'll do it all the time. Rarely patients choose that route because it doesn't stop the disease. You'll progress. You'll have no motor movements. You'll be what we call in medicine locked in. You might move your eyes a little bit. You can use tracking software nowadays to communicate through a computer, but you're completely dependent upon others, and it's an extremely expensive option that very few patients. The Hopkins Clinic for ALS is one of the largest in the country, and of the 400-odd patients new patients we'll see every year, maybe only a handful choose to be on a ventilator. So it's a fatal disease. So ALS is not a common disease. There are only about 6,000 new cases a year. In the US, we estimate that there might be 25,000 to 30,000 individuals affected by the disease. We'd have a lot more if we kept patients alive longer, because patients live so short. We don't accumulate a population. If we could keep patients alive, this number would rise quite dramatically to hundreds, many hundreds of thousands of patients in the US. The telling number is that 1 out of every 500 deaths in the US are due to ALS. That's really common. I guarantee you almost everyone in this room will have a friend, family member, neighbor who'll develop this disease and will go on to die, of course, of it. But it is part of a larger family of diseases. ALS is part of a family of diseases of neurodegenerative disease, which includes Parkinson's and Alzheimer's, both of which, like ALS, are largely sporadic, meaning they happen out of the blue. There's no strong family history. And as you'll see soon, we're learning clues about ALS that tie it back to larger populations, a rarer dementia known as frontotemporal dementia, and even maybe Alzheimer's. And the real clue came that changed the view of ALS. So as I mentioned, ALS is largely sporadic, no family history. About 5 to 10% of the time there is a family history. And in medicine, not just the neurosciences, but medicine across the board, we've really made advances by understanding and studying those rare subsets of patients where you can pin down why the disease started. This gets back to Steve's introduction about IBBS and fundamental neurobiology. Great advances, ultimately the advances to patients, often relate to those fundamental basic science discoveries, whether it's a new gene, the mechanism of a new gene, and that's certainly what's happening in ALS, and I'll tell you about that briefly. So the world of ALS changed because on the pie chart where it says familial ALS, got to get my pointer. In 1992, the first gene was described, and that was superoxide dismutase. This is an antioxidant enzyme. You know, antioxidant stress is all the rage in all of medicine. And this antioxidant enzyme, which is present in every cell in your body, yet mutations of that single protein causes this disease called ALS, a disease of not all the cells in your body, only of these rare motor neurons. And for many years, that was the sole mutation we knew about. I'm not going to spend time talking about the other mutations, but basically half of this pie was unknown. We knew that SOD1 accounted for about 15% of familial ALS, never present in sporadic, again, the vast majority of patients. But about 2.5 years ago, 2 labs, Brian Traynor, partly out of Johns Hopkins and the NIH, and Rosa Rademacher, who's down at the Mayo Clinic in Florida, simultaneously discovered a new gene, a gene which has a horrible name, and to this day it still has a horrible name, called C9orf72, which really means chromosome 9, open reading frame 72. We don't know what this protein does. The gene codes for a protein. We don't know what it does. We still don't know, and it may not matter, actually, and I'll explain that to you in a moment. But this gene had a very abnormal mutation, which I'll show you in a second. The first important part of this gene mutation was that it accounted for almost half of all familial ALS. Wow, that was a big chunk of patients. I mean, now, in fact, there's only a little bit of only a handful of patients we don't know in the inherited form that we don't know about. The unexpected part was that up at least 10%, and in some countries in Europe, 20% of patients with no family history, nope, mom and dad didn't have anything, uncles and aunts and grandparents didn't have anything, no family history, carry this mutation as well. So suddenly a mutation, instead of being a rare, although important mutation like SOD1, a major inherited mutation, but actually a major part of this pie. This pie had nothing in it before, just sporadic. So that was really unexpected. Okay, so 2 unexpected things. Big chunk of familial, sporadic. But wait a minute, it wasn't just ALS that had this mutation. As it turns out, rare patients with ALS develop a very severe dementia known as frontotemporal dementia. In fact, Dr. Hillis, who's in the room, is an expert at that. So a rare dementia has nothing to do with the motor system, and rarely you'll see patients, I'll see a handful of patients every year, that have this bad dementia and ALS at the same time. It turns out this mutation, you could take everything from this title and say this is frontotemporal dementia, which is a relatively rare dementia, about the same numbers as ALS. At least half of frontotemporal dementia carries this mutation. At least 10 to 20% of sporadic frontotemporal dementia carries this gene. And the real kicker is that you could have a brother that has ALS and a sister that only has dementia. Same mutation, same family, very different clinical diseases carry these mutations. So suddenly this mutation turns out to be quite common. In fact, we now know in larger population genetic studies, this mutation is actually found in some patients with Alzheimer's disease and some patients with Parkinson's disease as well. We don't understand that yet. but it's clearly a lot more than just subsets of ALS patients that carry this mutation. And in fact, ALS, there are other mutations that have been studied, but the connection with C9 becomes a major connection. By the way, I'm not going to say C9 or F72 anymore. I'm just going to say C9. It's much easier. Well, it turns out if we go back a step, when we as a community have studied neurodegenerative diseases over the years, especially in the 90s, the 90s was the decade of the gene. Many genes were discovered. Those discoveries laid the foundation to understand what starts a disease. And a lot of our basic science colleagues, although I consider myself also a basic scientist, started using those genes and putting them into mice. And it turns out in all of degenerative diseases, Alzheimer's, Parkinson's, Huntington's disease, spinocerebellar ataxia, there's a whole long list of these named diseases. The best model then and today is still ALS. You put an SOD1 mutation in a mouse, this mouse develops a disease just like humans. Develops slow weakness, can't breathe, and dies. Reliably. None of the other models nearly look as good. And that's great. It allowed us to understand the pathways of the disease in a mouse. So it's now about 25 years later since we built that SOD1 mouse. The good news is it really is a nice model. It allowed us to test a lot of drugs. The bad news is we use it to test a lot of drugs with great hopes that this would be a fantastic tool to find new drugs for patients, because I'm going to be covering the range of mechanism of disease to therapeutics. And unfortunately, this is a really crappy mouse to find drugs in patients. And it took us 2.5 decades. No fault of ours. It takes time to build through science and test things, and especially when we try to translate from a preclinical tool like a mouse to humans. Human trials take a long time in any neurologic disease, certainly any degenerative disease. And this mouse model has just been a failure, unfortunately. A great tool, not a failure to understand mechanisms, but a great tool to really find drugs. So we have to find other ways to do that. And I'm going to come to that in a minute. Nevertheless, this was the starting point in using genes to understand pathways. So it turns out the other last kicker, and I'm going to spend a lot of time around this, is this mutation. This is not an average mutation. You heard earlier, so I'm not going to repeat a lot of the kinds of things we think about when we see a protein and an amino acid can be distorted in a protein or missing, and then that can cause the enzyme or protein not to work well. That's often the basis, or can be the basis for some diseases. In this case, the mutation was not in what's called the coding sequence. It was in the introns, the sequence that don't code for a gene, in this particular gene C9. And it turns out the nucleotides that make up all of the coding sequence for DNA, sometimes they get repeated in diseases. In this case, this repeat, which is known as a G4C2, guanine and cytosine repeated 4 and 2, thousands of times extra. Think of it like as a big mole that grew the size of this room. Thousands of times extra. And that's shown sort of schematically. That would be the normal. All of us actually have a normal amount of repeats of this particular intron of G4C2. All of us do. Up to about maybe 20 or 30 repeats. That's normal. These patients will have thousands upon thousands of these repeats. And that big expansion is sort of shown here. This is the normal gene, normal gene. This big chunk of extragenetic material seems to be the heart of this disease. I'll mention that frontotemporal dementia, which is also concomitantly seen with this mutation, is the second most common dementia that's seen. It's a little different than Alzheimer's. It has a little different part of the brain that's affected. There's dramatic language abnormalities, the ability to produce language and speak properly. And this, as I mentioned earlier, is a common mutation. So the key, once we found this mutation, was to understand why is it causing disease? What tools can we build around that mutation to understand the disease and eventually find drugs for the disease, and tools so we can go back to patients to use those drugs, often referred to as biomarkers. So right away, once this mutation was found, there are theories about what this mutation could cause. So if this is the DNA eventually coding for RNA, these expansions tend to wrap around themselves like layers, and they're called G-quartets. They layer and layer and layer and layer and layer, and that's shown schematically here. And it's believed that that layering alone that can interfere with just the gene being made into protein. So one theory was that this protein's not really made properly. Probably the more prominent theory is when you have and I'll again be going over this, when you have this big expansion of material, it aberrantly sucks up other proteins in the nucleus of a cell. Think of it as like flypaper. The flypaper, if you hang it out, it's going to just stick to a lot of things will start sticking to it. Or the thing that you remove lymph cat hair, at least I use it to get cat hair off my pants. And so this big expansion can bind up proteins inappropriately and suck away other proteins in the nucleus of a cell. That's called RNA binding sequestration. Don't worry about the name as much about it. It's a mechanism by which this big expansion can disturb a cell. And lastly, a brand new biology that we never knew about until neurologic diseases is sometimes this big chunk of DNA can leak out into the cytoplasm of a cell and aberrantly form proteins. And it's called RAN-associated or repeat-associated proteins. And all of those were believed to potentially be occurring in the disease. Now, as a background, we don't know what C9 does. Again, it's a gene that has multiple introns, of course, and exons. This mutation is found in the first intron. Less important for today's discussion. We don't yet know. We have some ideas about what the protein may do, but many people don't really think that what the protein normally does is really that important. Now, what do we do? How do we try to understand this? So typically when you have a mutant protein in today's world or a mutant gene, you try to build a model. You might try to, and it's very common to build a mouse model or a fly model or a little fish, a zebrafish model. Well, it turns out this humongous expansion of DNA makes it really difficult to make a mouse from. You can't, the normal molecular tools that we do to clone genes won't work very well for this expansion. And we learned about this expansion about the same time we were building new tools, and Hung-Jung Sung at the beginning told you about those kinds of new tools that we use in neurologic disease today, or many diseases actually, and those are induced pluripotent cells. So I'm not going to review for you what induced pluripotent cells is. I think actually many of you in the audience have written about stem cells and how we derive them from skin. In this case, we can turn them into brain cells, brain neurons, and the support cells of the brain known as glia. As it turns out, and I'll skip the slide on what IPS cells really are. We were fortunate in that when this mutation was found, the NIH, and the members of the NIH here today, they were prescient enough to recognize the value in these cells as an alternative tool, because these aren't mouse cells. These are human cells. These are derived from the very patients I see every Monday in my clinic. They contain the natural mutation, not some artificial mutation we stick into a mouse. So maybe, in lieu of all the problems we've been having with mice, maybe we should be turning to use this as a tool. And you heard Hung-Jung Sung describe to you how well they've been using it in schizophrenia. Maybe we should be using these tools to understand the disease, especially because cloning this mutation would be just a humongous task, would likely fail. And in fact, it's been going on now for 2 years, and it has been failing. It's been very difficult to clone this, a big chunk of DNA, and put it into a mouse. Very challenging, and every time you try to make it, the animal's cells normally chop it up into little bits. So it's not working well. But the human cells are a great possibility. So it turns out NIH had funded a library in 3 different disease areas, in fact: Huntington's disease, Parkinson's disease, and ALS. And I run the ALS consortium to build this library of IPS cells so we could not just use them locally at Hopkins, around the world. In fact, they've been shared through many, hundreds of labs around the world, these cells as a platform to study the disease. So we shifted. Now, the key thing is, again, going back to the 90s, we've been using mouse models for a while to study the disease. And again, not just in ALS. Mice look like ALS, but are they really human ALS? Because if you're trying ultimately to translate something, a mechanism of disease or a therapy, you better darn well be sure whatever that tool is, if it's not your patient, it really represents the patient. So if we were going to use these cells to study the disease, the first and foremost question had to be, do these cells accurately reflect what we know occurs in humans? And so I'm not going to go through everything on the slide. This slide is a verification that, well, we know this disease, this big expansion can be found in humans, the gene expansion. Do the cells show that same gene expansion? So this blot, don't worry about what it shows, says yes, it does. The cells really look just like the human brain. By the way, we can't take brains from our patients, so when we look at human tissue, it's always from autopsies. Other disease areas, non-neurologic disease areas, they have a field day. They can do biopsies of liver and pancreas and skin. We can't do that in neurologic diseases. It's one of the challenges in developing drugs for the central nervous system is that we can't sample from our patients. We have to use these surrogates, or we have to use autopsy material. So the cells have the same genetic abnormality. Well, next, do the cells actually have a loss of the RNA that codes for the protein, was what had been predicted? Actually, in the brain, there's about a half amount of protein and the gene actually, and the cells are identical. So the loss of the gene is in the cells the way it is in the human brain. Well, there's a pathology. When this big expansion begins to accumulate in the nucleus, one of the first things we began to find as a research team was that you get foci of these RNA. All this extra RNA forms these foci, pathologic foci, with the assumption that these foci mean the cell's not doing something right. We can find those foci in the cells, and we can find it in the brain tissue of our patients. So the same pathology. The presumption is that these foci disturb how the cell processes its DNA, or more importantly, the RNA. Can we find that in patients? Yes. We can screen for thousands of RNA changes in our patients, and it turns out the cells match that beautifully. So just on this slide alone, it's taught us that the cells have almost the same identical changes that a human being has. Now, they're human cells. So that speaks pretty well to them being a reasonable circuit to use as a tool. And lastly, I mentioned that in a way that we don't fully understand, this RNA can leak into the cytoplasm of a cell and make a peptide, a protein known as RAN protein. We can see that in the human brain. Only ALS patients with C9 have this unusual protein in the cytoplasm. The cells have it as well. We have 5 different measures by which the cells completely match what we see in human brain. We've actually never seen that in mouse models. I don't mean for C9, but in general, there's been evidence that mouse models, and by the way, I'm not trying to dig on mouse models, but it's a new world. We have to find better tools to find drugs, and these cells are really nicely matching what we expect. So they recapitulate. Well, okay, so they look like the brain. Can we use them as tools to understand maybe some biological processes that are defective? In part to understand the pathway of disease, but actually, probably more importantly, as a tool to screen for drugs. Because if these cells are going to represent what occurs in my patients, can I use them as a platform to find drugs that fix a lot of those properties? Well, it turns out, don't worry about what the slide says, we can find the glutamate, which is a well-known chemical in the brain that allows you to see me right now, but when dysregulated, kills cells in the brain. These IPS cells, these neurons from our patients, highly sensitive to glutamate. So a readout. The endoplasmic reticulum is part of a cell that controls how proteins are made, ultimately. And it's well known that in certain disease states, that process can become distorted, and you can press that process. You can stress it in something known as ER stress, or endoplasmic reticulum stress, by using drugs. It allows one to see the phenotype of a highly sensitive cell. These cells also show increased sensitivity to what's known as ER stress. Again, not as important about what that is. What it really means, it's a readout. We can use these cells to say, okay, they're more sensitive. Will a drug abrogate that eventually? And I'll get to that. So we have a series of events that occur. The cells match the brain. There's some stress readouts. What does it really do to begin the process to kill a cell? What's the process by which it might be disturbing the cell? And here we can take lessons from other biology of diseases where these expansions occur. So Huntington's disease, and there's a variety of neurologic diseases that are actually not well understood, but have similar expansions. And if we look at this cartoon, this expansion is sort of schematized here. I'm pointing only to this side. Tends to bind up, or it's been theorized to bind up, other very important nuclear proteins known as RNA binding proteins. These are the proteins that eventually carve up the genes, allow the genes to individually work, and ultimately create new proteins. And when you have this huge expansion, they sequester. They just suck up all these RNA binding proteins. So if you normally had 10 in the cell, they have to keep working. Just this expansion alone might suck up all the 10, and the cell has no more of those RNA binding proteins to do what it's supposed to do. And so the expansion essentially depletes the cell of an important protein. What do we do about that? Well, there's a therapy known as antisense oligonucleotide therapy. It's the equivalent of a gene therapy where you can take very short segments of genetic material. It will bind to this expansion and prevent all those proteins from binding up. Take your fly paper, throw a lot of other tissue paper on or a powder, and it won't stick anymore of the flies. And that's what's done. And it can be done both in cells, and it's actually already being pursued in patients, not for ALS yet. So it became a very feasible therapeutic approach to think about using ASOs, or antisense oligonucleotides, eventually. Well, first, what are the proteins that might be being sucked up by this big expansion? Because that allows us to understand the events that occur because of the expansion. And with a group of colleagues at Hopkins, we screened a variety of proteins that might be sucked up. And we found a series of candidate proteins that might be abnormally removed from the cell. And as we looked at these foci, and tried to understand what those aberrant proteins are. I'm not going to go through that list of proteins. We've studied them and found some that are very important in the normal clipping of DNA. But as we looked under the microscope, trying to understand what the defect was, we noticed that these expansions, as shown as these red dots, sometimes were located at the edge of the nucleus, the perinuclear region. In fact, we looked at even other publications where people weren't paying attention to that. We saw repeatedly they tend to be at the edge of the nuclei. And in fact, a group out of the UK very recently quantified that and said, gee, about 30% of these foci tend to be at the edge of the nuclei. So why were they there, and is that in any way related to the problem? Now, if I go back one slide, I probably should have done this earlier. When we looked at the list of things that bind to this expansion that could be inactivated, again, like the flypaper, one of them that came out on our list was a protein known as RAN-GAP. This is a critical protein in what's known as the nuclear pore. I'll come back to that in a second. So we had these series of proteins that were abnormal, and we turned to a colleague to say, well, we're going to try to study these proteins, but you have a fly, and this fly actually has the mutation. We did build, or others have built a fly, and this flies, when they have a mutation in a human gene, sometimes the easiest thing to look at is their eye. And the eye gets all, it's hard to be seen, it's hard to see in this light, but the eye becomes very irregular and degenerates. So it's a degeneration phenotype. And actually, the flies might not even live as long when they carry this mutation. So the fly can be used as an alternate tool, not a perfect tool, but an alternate tool to find the defect. So we went to our colleagues at Hopkins and said, we've got these genes that seem to bind to this expansion. We'd like to understand which of them could be somehow related to the disease. And so they took all of those genes, looked at them, and found, when we look at all these other genes that could be binding, a handful of them made the fly eye a lot worse, and a handful actually improved the fly's eye, which could give you clues about the mechanism because they're essentially being therapeutic. And an example of that is when we look at the fly's eye, again, in the light you may not see it, here's a normal fly and the elements that make up the actual photoreceptors of the fly, and you can see them here. But in the ALS version, the C9 version, after about 2 weeks, that's all disorder. The eye looks abnormal, and a bunch of these photoreceptors are degenerating. But if we fix, by overexpressing one of those proteins I told you about earlier, this nuclear pore protein called RAN-GAP, we completely fix the fly's eye. That suggests that maybe this pathway, this protein on the nuclear pore, could be important, this RAN-GAP protein. So, okay, that's a fly, though. What about going back to our human cells that seem to so nicely match the pathology of humans? Is there a problem in this nuclear protein? I'm going to tell you more about the nuclear protein in a minute. Well, in fact, when we took the right tools and started looking at these cells, we saw, oh my God, there's big clumps of this nuclear protein, this pore. The nucleus has pores in it that allow things to go from the nucleus to the cytoplasm, and it's all clumped up abnormally in the C9 patients compared to control, and that's shown here in these big dots. And controls really don't have it. C9 have an aberrant amount. And if we look closely at this, you can see this protein is much larger than normal and huge clumps of it poking through the nuclear membrane. That's just the nucleus of a cell. And furthermore, when we look closely, it's not just that the protein's abnormal, it's all clumped up with this expansion RNA. So the abnormal expansion is clumping to the cell nuclear pore. That's the IPS cells. Do we see that in the human brain? You bet we do. In some early studies, we can find the same kind of problems. Again, IPS cell and brain. So what's a nuclear pore? So nuclear pores are one of the largest protein complexes in our body. They surround the edges of the nucleus. They're made up of many, many, many proteins that all come together, and it regulates what goes from inside the nucleus to back into the cytoplasm, but importantly, it also regulates what's in the cytoplasm, because the nucleus has to have proteins, so what's made in the cytoplasm of a cell that gets back into the nucleus of a cell. And they have this almost science fiction gross-like structure. I'm going to show you a video in a minute where there's filaments that grab some of the proteins, throw them through this basket, and throw it back into the nucleus, and vice versa. So proteins can go either way. And RAN-GAP is right in the middle of this pore. It's right on the edge of this pore, and it's critical for moving proteins in and out of the nucleus. The very abnormal interaction in ALS, the very protein that's all stuck and aggregated in these nuclear pores, is critical for the movement. And if we look at what this actually, how it works, this is a video, YouTube's fantastic. This is the nuclear pore. and it dynamically captures individual proteins or RNAs and pulls them through and back and forth. This is not from humans, this is a yeast equivalent, but there's a huge conservation between proteins made in yeast and low animals as well as humans. This is a critical fundamental process in all of our cells. And now we're moving into the nuclear pore, and there's this meshwork, the FG meshwork, that sort of acts almost like a filter to handle the movement of RNAs and proteins between these two compartments, two critical compartments of any cell. And that's what you're seeing here. It's bound, goes through the meshwork. I said this is really when I saw this, it's very science fiction-like. You're now that element going through the meshwork, and pretty soon you're going to be thrown out. And what it appears so far is that our C9 expansion is chunked up. It's stuck into this meshwork and in some way disturbs it. Well, I said disturbs it. I haven't shown you data of that. And so, in fact, when we go back to our fly, oops, I'm just going to skip through this. We can use the flies, again, as a dynamic tool. And I'm not going to really go into detail other than to say that if we label proteins with green, we know the proteins normally are both in the cytoplasm and the nucleus. And this is the nucleus. That's the cytoplasm of these salivary gland cells in the Drosophila fruit fly. But in the ALS version, you don't see protein making inside the nucleus. It's not being imported properly. There's a lack of import. So again, controls are here, but in ALS, you don't see very much green in the nucleus. And in fact, the larger the number of expansions, the lower the amount. So here there's only a few repeats, and the nucleus is pretty much clear. It has some, this is actually mislabeled, it's the opposite. From my angle. Basically, the larger number of expansions, the less the protein gets into the nucleus. It appears to interfere with the trafficking of proteins inside the nucleus. So now we believe, again, I've been relatively short in what I've been telling you, now we believe that the C9 expansion, these big chunks of these G-quartet structures, aside from binding up other RNA binding proteins, they're getting stuck in the nuclear pore and they're preventing the normal traverse of proteins or RNAs inside and outside of the nucleus. If that's all very true, then that antisense therapy I mentioned to you earlier, if we coat this expansion with antisense oligonucleotides, we might prevent it from being sticky and prevent any problem from occurring. In fact, that's what we did. In collaboration with ISIS Pharmaceuticals, probably the best company in the United States. I don't get any money from them, by the way. Although I wish I did. NIH is good, but they're low on money. ISIS has now screened 400 different of these oligonucleotides. They're very short segments of a DNA sequence. We screened 400, we narrowed it down to about 10 or so, and we then used them in our IPS model to say, I told you about all these defects, about those aggregates that form, the glutamate toxicity, the ER stress. With these molecules in this human cell that, again, has a human phenotype of the real disease, will those drugs work? And the answer is, you bet. We can get rid of those dots, those RNA foci. We can block glutamate toxicity. And we can even sometimes interfere with those dipeptides that are formed in the cytoplasm. So we can use a human IPS cell, and Hung Jung mentioned this earlier this morning, to invalidated cascades, pathology that we see in patients, now in a real human cell to show these drugs work. That's a fast track. This would normally take a long time. These experiments took 2 years. We're already working now on developing that ASO, those oligonucleotide therapies for our patients, with the hopes within a relatively short period of time, I won't give you a timeline, it's a mistake to tell reporters that we'll go to patients hopefully soon, because this is a very targeted molecular approach to this kind of disease. Not only do we fix all the pathology, I told you there were genes that were up and down, upregulated, downregulated in the human brain. Did we fix those genetic aberrations in the IPS cells? You bet we did. Don't try to read this slide, but this slide basically tells me to tell you that, yes, genes that were aberrantly turned up because of the mutation, now they're back to normal. Genes that were low, not expressing properly, now they're back to normal. So we can have a dynamic readout. And those genes, because they're normalized and because they're real readouts, they might be biomarkers. Because, and I'll repeat this in a few moments, one of the critical things that when we go and develop drugs for the central nervous system is Are the drugs going to work in the CNS of our patients? What evidence do we have that the drugs actually get in to the brain? This is something that's very easy to do in heart disease and lung disease and kidney disease and pancreatic disease, because you either biopsy the tissue, you measure blood, but in the brain, when I give a drug to a patient, I cannot take a part of their skull, take a piece of brain afterwards and say, yep, that drug's working, or yep, that drug got in properly. Sometimes I can do spinal taps. So this gives me the clue of what I actually might be able to measure in the CSF, the spinal fluid. You all probably know what spinal taps are in our patients. And in fact, we can test those drugs, not just in the IPS cells. We can even show that in our fly model that we can fix that nuclear import trafficking. So we know in a dynamic, a little more complex organism, it will work. So the ASO treatment, the ASOs, the antisense oligonucleotides, rescues the animal from dying and fixes that movement of particles into the nucleus. So if I step back, because I only have a few slides left and it's getting late, one of the great problems we face in brain sciences is that there's a real trouble developing drugs for neurologic diseases. Many other diseases have a higher success rate, but neurologic diseases, especially chronic degenerative diseases like ALS, Alzheimer's, and Parkinson's, where it can take years to see if a drug slows the disease down. We certainly like to cure the disease, but generally we're trying to slow the death down. By the way, as a side note, remember, the great advances in cancer, of course, and we've seen great advances in cancer, come from looking at rare subtypes of the disease, knowing the pathway, finding the right drugs. But most importantly, especially when you're a neurologist, it's all about poisons. Because what are anti-chemotherapeutics? They're poisons. You're killing cells in many cases, not always, but in many cases, you're killing. In neurologic diseases, it's the 180-degree opposite. We don't want to kill brain cells. We have to keep them alive. And keeping cells alive is an enormous challenge in drug discovery. So you want to be more sure that your drug, yeah, it's getting into the brain. You have a readout, some biomarker. I give this drug to a patient, the drug is really working. So in neurologic disease, we do have a high failure rate. In fact, we would love as we go from a preclinical work to a phase 1, 2, and 3 trial that we have a much higher rate of success. And yet in neurologic diseases, the failure rate, even when you have a phase 2 trial, the first trial to show efficacy, that almost 70% of phase 2 trials fail in phase 3. And they often fail because companies have not developed the tools to know if the drug gets into the brain and it's working at its target. Now, why did I go off base for a second to tell you that? Because if we're going to do this properly, we as academics, and we're going to try to advance our understanding of, in this case, dementia and ALS, and develop this pathway into a drug, at the end of the day, I need a marker. I need to know that I have a tool that's going to be useful to tell me the drug's working. Again, this is where IPS cells are fantastic. So I told you before that one of the byproducts of this mutation is that this peptide's made, this RAN dipeptide. Now, we don't know much about it. Some people think it's in and of itself it's toxic. I don't, but it's an ongoing theory. But it's this dipeptide. It can be measured in the human cells. Can't do that in a mouse. They don't make the peptide. And the human cells leak this peptide. So we can use the cells as a readout of what our biomarker might be, which is this dipeptide, this GP dipeptide. And actually, we've done that. We've done a very small sampling already of CSF, spinal tap from our ALS patients, and lo and behold, that this dipeptide is massively increased in patients with C9. It's not present. That's a good thing in patients that are healthy. It's not present in patients who have ALS who don't have the mutation. So it's a great readout. And what I'm not showing you here is that some patients, just before they get disease, we already have a clue of this, just before they get clinical disease, they come to us, they sort of look normal, because clinical exam has its variances. Do a spinal tap, the dipeptide's elevated. Several weeks later, they develop the disease. So it's an early marker of disease, and it could be a fantastic readout for as we develop the drugs, we're right on board with a way to measure that the drug's working and to determine early on who are the right patients to bring into trial. Now, antisense is not the only approach that we can use in the last minute. There are small molecules, chemicals, because antisense is a brand new therapy. In fact, the only other neurologic disease that's been tried in was ALS, the SOD1 mutation. We ran that trial at Hopkins and 2 other sites and showed that we can deliver this therapy. We don't know if it's going to work yet. It sets the stage for using this novel approach to gene therapy for a neurologic disease. But small molecules have been studied for years to unbind this G-quartet structure. And others right now are finding small molecules might unbind this and also be effective. So different ways in which we would envision approaching ALS. And again, this is not just ALS. Although I've used this talk to use as a platform for ALS, this mutation is found in frontotemporal dementia and other dementias as well. ALS is just a good starting point. And lastly, if we're going to be effective at doing clinical trials, the other end has to be done well as well. As much as fundamental neuroscience or cell biology is the foundation by which we eventually bring therapeutics, that other end of the coin, the clinical trial, it's equally important. If I have a drug and I don't run that trial properly, or I can't bring patients into my clinic except for one a year, I won't be able to actually test that science out and test that therapeutic. So to be aggressive about this particular disease in collaboration with the NIH, there's actually C9 clinics starting to bud around the country that'll not only identify this subgroup of patients, but be primed and ready to bring them into trial in an expeditious manner. Because, and as my last slide, CNS, even though this is really about dementia, CNS therapy development really means the future of, or at least I believe the future of CNS therapy development really means understanding these subsets of patients, not treating ALS, or for that matter, Alzheimer's and Parkinson's patients is all the same. We have failed for decades doing that. It's time to take the lessons from cancer therapy, and instead of treating everyone the same, go after the subsets of patients where you really understand the mechanism, you have readouts for drug action, you know if your drug's working by having these biomarkers, and yes, I will not likely be able to treat all my patients, but I've failed, I, meaning representing our community, for doing that for years. Begin to target the subsets and beginning to get traction where you really understand, again, starting from the fundamentals and bringing that up to therapy development. Many pharmaceutical companies are now beginning to see this approach at least in neurologic diseases. And now we're embracing, instead of going for all patients, a very coordinated, a very well thought out process of developing the biomarkers, understand the molecular pathway, and go after those patients. So the work I showed, Chris Donnelly is actually a postdoc in my lab who did just a phenomenal postdoc. If you were academics, I would telling you to hire him. I do not want him to go into your field, I'm sorry to say. I want him to go into an academic position and continue the great science he's done. But all the work that we do at Johns Hopkins is extremely collaborative. And I failed to mention the many people who have been involved in the projects that I talked about, which span both the basic science, basic cell biology, as well as my other clinical colleagues. And all of what we do, of course, requires funding agencies. And the top of this really, of course, should be the government. NIH funds the vast majority of research, but we have other nonprofits that act to support us. And I'll stop because it's lunchtime, I think.